Start recording. All right. Thank you everyone for joining today's seminar from IOB. Um, this is the first virtual seminar of this spring semester, and we are joined by Dr. Thomas Otto, who is joining us from 6 hours, I think, ahead. And Germany, it's like I think 5:35 at his end. And that was something actually he figured out yesterday, if I remember correctly, because like I think the daylight saving time just 5 hours, 6 hours, it confuses. But that makes it like a great honor for us that he's joining us from that far away. Dr. Otto has been working on the field of computational biology with a focus on infectious diseases, and he has been a very important person in understanding pathogen genomics, assembling pathogen genomes, annotating them, and so on. And he has actually worked all over the world because if you look at his CV, he actually started working in Germany, then moved on to Florida, so came to USA, then did his PhD in Brazil, then he worked in Wellcome Sanger Institute, which probably a lot of us have known by name. Then he was affiliated with University of Glasgow, where he's still a professor. And finally, I think just last year, very recently, he has joined the Bernard Nocht Institute of Tropical Medicine that is back in Germany. So I think in a way it's a circle. And I remember circles because he was involved in a tool called Circulator, which I have been using in my project very recently. And uh, Jesse, my PI, is on this seminar, but she knows how important it has been to this. And that's how I got to know his work. But he has been involved in a lot of tool development as well as analysis. And I think in IOB, we have people from very different backgrounds, but we all try to make sense of data in one way or another. And that's where his insights will be very interesting for us to learn about. And I'm very excited to know what he has been working on. So please have a warm welcome for Dr. Otto, and let's see what he has in store for us.
Thanks, Rafid, for this nice introduction. Can you all hear me? Yeah, good. So, yeah, thank you for inviting me and having me over. I nearly missed it. I had in my calendar my time an hour later, but luckily I checked. So we are. So today I'm talking about resolving gene regulation in cellular heterogeneity. It's a long title. I wanted to call it just novel tools, non-computational tools in computational biology. And then I just asked ChatGPT why things, and it told me off. It said, make it more interesting. So if the talk is not good, it's not my fault. If the talk it works, then we know here we are. So I tried to today give you a bit on my overview. I just gonna quickly talk a bit about the Bernard Institute for Tropical Medicine, talk about a bit about the regularly cellular heterogeneity malaria, and then I'm going to talk about 4 tools have developed and now in the end phase of just being published or going through rebuttals. And it's a kind of different talk. I try to give you an idea rather than going too deep in the math or in the biology, but mostly trying to pitch where the issues are and why we're doing those tools. And in the end, often you read those tools like Circulator, and if you think about it, it's a simple tool, and the way it solves it is easy, but it's just easy once you know how to solve it. And that's something that is always interesting to think. So here I am. Or now it's a bit later. This is a Bernard Nocht Institute with the new building, the old building, and this is basically the old hospital, and I'm kind of below here now. It's very close to the harbor. It's a super nice building, but the question is why I moved. I think it's good to move. So every 5 to 7 years, as an academic, if you can. I think I had a professorship in Glasgow. I liked it a lot, but then I got this unique opportunity. But now I think now is good. Now I will stay here. At least I tell it now. The Bernard Nocht Institute itself. That's a bit of German, but below you see the translation. I think what is very nice is just around 400 to 450 people. It's quite small, but it's kind of embraces One Health idea quite nicely. So we have the pathogen part. So starting at the point of intervention, then how do the pathogen work with the hosts, immunology, patients, intervention, and then population and implementation. And I think it's nice because it's kind of compact. It's not a big university. I mean, Glasgow, it's a great place, top 100 university, big thing, but here it's smaller and very much nicely connected to the global south. So a lot of our clinicians, when you have chats with them, they're mostly actually in Africa somewhere. So we're really nicely connected, which is quite nice. And then it's broken up in those different well, departments or groups, and you can see it's a kind of very vertically structure. And the idea is for my data science center where I'm part of is where we horizontally bring it together. So the idea is to support with our tools then all the different parts of the institute, and we do that through collaboration, through teaching and interaction. If you are interested, maybe we can chat about this later. So going now a little bit about my pathogen of interest, although a lot of these things I'm telling you is not just malaria specific, is that, and I haven't like the normal life cycle slide with all the different life stages, not really relevant here. I think important to say is that it's very heterogeneous. So if you take one infection here, you have a single cell life cycle with gametocytes. It's we often take single cell. It has a lot of dropouts. It's expensive. It's noisy. Can you apply it in the field? So often it's very difficult. I study basically gene families around VAR genes that are important. So they're basically on the infected red blood cell, and they're basically anchoring and kind of site adhered to the blood vessel. So basically they hold to then not get basically pulled into the spleen where they basically would have then been cleared. And when you have that, you can see here. You can see a blood vessel in the brain, and you see the red blood cells there. And basically here you have the parasite, basically a site adheres, and with that you get the blockage of the brain. With that you get brain malaria, and you get this high numbers of death, which is, I'm gonna give you a number, which is kind of 600,000. And is it a lot? Yeah, so it's kind of every 40 seconds a child dies. So there will be, if you put it in the time I speak, there will be 60 kids that would have died from malaria. It's still a huge disease. But there are other things that's complicated, like the parasite kind of site adheres in different tissues. So it's in the brain, it's in the bone marrow, where it's important for gametogenesis, for development. So basically, we need to deal in different fronts and with different facets of data. The first one, and what I try generally is to kind of build tools to kind of answer this question and try to dissect these. And with a general focus on host parasite interaction. And the way we basically approach that mostly in my lab is we have the different projects. It's not just malaria, it's also trypanosoma cruzi, another parasite. We also jumped onto the COVID train, there's so many. And basically we're interested to kind of to measure, and in the past we basically did a lot of sequencing. I guess you all know bulk sequencing. So, but bulk, you just basically take here, for example, the whole tissue of the brain, and then what you just get all it together. So with the advance of single cell that you probably know all, you can now actually sequence individual cells. You can form that to functional units and cell states. And then you can see, oh, we have this new subtype, we have this parasite behaves differently. But what I'm really interested is how do then these different units communicate? So is it like if it's an orchestra with the director who says, you play this, you play that, and oh, look, there's a parasite coming drama? Or is it more something chaotic, like a jazz band where something is coming and then this musician reacts to it and then the other follows? So immune cells cascading signal. So that's basically the other way we would try to understand. The way we do this, we do it with single cell, and now we also do spatial transcriptomics, which I mentioned very late in the talk. But most importantly, as so many things, we need algorithms. We need ways to generate the data, to quality control the data, and to visualize the data, and then help afterwards our colleagues to analyze it. So the first project I'm going to talk is about the so-called VAR gene family, which I mentioned, the one that's responsible for the site adherence. I basically just put PFMP1 into Google, and that's what it told me. PFMP1 is it's a plasmodiaphysated parameterocyte membrane protein 1. This is basically coded by the VAR genes. It's a very highly labeled gene family, roughly 60 genes. They are pretty much basically 60. I assembled many of them. It's on the, as I said, surface. And very interesting, it has this multiple exclusive expression. So it seems that in general occur, there's new work now, but overall there's on one parasite just one vargine expressed for the cytidine. It doesn't make sense because you don't want to show all your repertoire to the immune system. Basically there will be antibodies and you just have 60 compared to other pathogens. So but how is it regulated? And very little we know, to be honest. Well, we know a lot, but in the end we know very little. So and if you look at the structure of vargines, what's quite interesting, the genome is very much structured the same way. So we sequence many genomes with long reads, assemble them. So I've said a big part of my work was actually to assemble genomes and basically get them together. And what you have the telomeres at the end, and then you have always one vargine that points to the centromere. And there's always a specific, we call it upsp. Then it's followed by upsace, and these are the subtelomeric vargine clusters. And then we have some, and it codes for 40% of the vargine, we have internal vargine clusters. And there you find generally upcs and some upbs. What are those? Well, if you look at the vargine here, it's a 2 exon gene. It has an intron, and on the 5' end, it has an upstream sequence. So around 600 base pairs, you can classify these into those 4 groups. And it's really, you can see in a second, you can really see it. And based on that, we can see the position. And basically with the position, we kind of know what is actually the phenotype. So we know that upsps, upsace are early expressed, more virulent, etc., where upcs generally are more to maintain an infection. That is something we're doing. So basically what we wanted is to kind of, can we detect this sequence? But why is that? Why do we need a tool to predict the upstream sequence of vargines? Well, because although you have that, in reality, you can't capture. It's very difficult and very expensive to do full genomes. And if you do a study where you're interested, where you sequence a lot of people, often you just sequence the tag region, which is 150 amino acids, cassettes, exon 1, or the whole gene. So without being able to sequence the upstream sequence. So what we want to be able to predict from the different sequences the upstream sequence. And the way you generate those could be tag sequencing with PCR primers, RNA-seq assemblies, or de novo assemblies. So the approach we took all the PacBio genomes that exist. There were 66 at that time. Got all the vargines together, built a tree, and added to that a very old Rust 2010 data set from Thomas Luston, where he annotated the up sequences, and then in colors you can see. And although I would say you shouldn't do trees of vargines, interestingly, it's quite stable for the upstream sequence. You can see here the genes upcs, upsps, upace and upsps. And basically we classified our true stat, and with that we did machine learning. So we took 80% to train and 20% to test. And and basically it's not really rocket science. You just load it, you train it. There's different ways to code it. And it was started with the master project of Aaron. And what you can see then here, and then we have different training sets. And overall, the accuracy looks quite nice. So we have an accuracy of 83%. It means that 83 times we're kind of correct. Obviously, the longer you input sequence it, the more precise you get. So shorter sequence is more difficult than longer sequence. We also had so the first 4 tools is support vector machine, is basically a linear classifier, and with different kernels, so different methods to approximate. And then we have a random forest. There's a different a machine learning technology, and then XGBoost is good for unbalanced traits. But overall, you could argue you see 5% of difference. It's not a lot difference. It's kind of all, so kind of we were, yeah, it's working. But is it all good? So first, there was another tool called CCAPS who was there before, and it works on hammer models. And we can see that overall, if we use single amino acids as entry point, we are similar in terms of overall quality. However, we realize that if we use longer input, so we always put 4 amino acids together, we get far better results. So really kind of in the better results. The other thing, okay, does it look good? Well, to be honest, not really. Because what we realized, we're very good to distinguish up-Cs, which is the group outside of the centromere. But but this, so if you, how you read this table, so this is for tag sequencing, the SVM linear methods, you could see we gave it 65, 67 ups-As, and it predicted all of them correct. If you give it 250, and basically we trained it, so we trained our machine learning methods and we tested it with a 20%, the ones you see here. From the 261 Bs, most of them were classified correctly as Bs. However, from the 66 Cs, most of them, 3 times more, were actually in reality ups Bs. And that annoyed us a lot. We tried a lot of things. It's unbalanced data set. But then in the end, we just capitulated because what is it? We just think it's just biology. It's recombination. So from an old Vargine paper, what you can see here, it's a circus plot. And basically what it means that this Vargine has, where you have the gray part of at least 2 kb, it means it's the same sequence here with here. So basically over its evolution, it recombined, it changed. And it kind of makes sense that genes that are closer together in the internal Vargine clusters, they recombine more because they anneal more together. And if they're together, and we know that they're Cs and Bs, they can recombine, and hence it's very difficult to have the correct ups sequence. But then we didn't give up because, well, if we have this high error rate, where are those mistakes? And most of these mistakes are actually in internal Vargine clusters. So basically it means that when it does something wrong, the other sequence is actually also an internal Vargine. So we changed a bit, we did a second classifier that we called a positional classifier, who basically tells us if it's an ups A, because we know that, if it's an ups B, subtelomeric, or it's an ups CB that is in the middle. And doing this, we could see we got similar values, which are also quite good, and we had a new classifier. But who cares, right? But the interesting thing is now when we applied that, so I guess we do care because it's interesting because we associate like internal Vargines more to be Vargines that are important for longer infections to maintain the infection rather than transmit very quickly. And we didn't have any bulk RNA-seq data, but we use an old data set we published some time ago where we basically have 11 countries. Each of them, we took 12 genomes and assembled the Vargines. And then basically, what we saw a pattern that when we order it by the numbers, by the relative number, percentage of CBs internal, by number, you can see that the genomes with less relative internal Vargines are genomes from countries in high transmission. Which is interesting in a certain way first, because it sees a strong pattern. But then it's very easy to make hypothesis to say maybe there are more subtelomeric Vargines needed in high transmission, because these are the Vargines that are important for high infections and then quick transmission. So basically, which is nice because we could use here our computational tool to see that we here have actually a bit of malaria hypothesis that we can take. So with that, I concluding the first part of the UpsML. So it's a tool that helps us phenotype Vargines or smaller sequences of it. It's still challenging. It's far away to be perfect, but I think it will be difficult to be better due to the fact that genomes and Vargines recombine. And just to say about the title, and we think that UPS sequence is important because as they are 5', they have a role in regulatory, in gene regulation. If you're interested, here's actually the link always to the GitHub repo. So now going basically from the gene regulation one layer higher to look at gene expression. And obviously, I heard about single cell. Some of you might have worked with single cell. And what we do a lot with single cell, we have two conditions and we want to look at the difference. So we have cell types from group A and group B, and then we do the pipeline, and we do differential expression. We have the gene that upregulated and the gene in one condition, gene upregulated in the other condition. It's good. We can find a lot of differences in heterogeneity, but how good actually are those methods? Are they really good? So what do I mean with that? Well, assuming following. That's a work actually from Yi, which is a PhD student in my lab. Assuming you have a ground truth of 100 differentially expressed genes. What you want your tool to return is to find exactly that overlap. However, if you look into single cell methods, what you then see, I need to put this camera away, if not, I'm going to look all the time at myself, which is not good. Basically, if you look at that method, single cell, overall, it finds nearly 1,500 genes. Of that, just 350 are basically true positives, and 24 it misses. So it predicts it has a lot of false positives, which are not differentially expressed. But it misses very few because it overfinds. Another method actually is called pseudobulk. What does it mean? So you put all the blue cells from each patient together in a bigger cell. So basically you put it together, and that's supposed to be the best methods. And if you look in the literature, that's now the Zimmerman paper, that's super duper, it corrects false discovery, right? So basically what you get, you basically have no false positives. However, you're missing 20%-ish of the real, of the data. And you could argue, well, that's not a lot, but this is simulated data. This is data that should be perfect. So we had an idea, Yi and Dom, what about, and actually this started as a datathon in the lab that a lot of people basically contributed, and then this then became Yi's master project. So rather than doing a pseudobulk, we basically just pull some cells together and generate meta cells. And with that, and with different methods to do that, we are still not perfect. However, we are, in this case, better than pseudobulk. There's some example in some simulation, it's the other way around. Pseudobulk is a little bit better, but overall, we always control the huge amount of false positives. But the thing is, like, this is all simulation. And what we know from single cell, it's really noisy. So we had this problem that we hope that our tool is good, but we didn't have the right way to test it, right? So how can we do that? So what we thought, we thought to use a real data set. And from a real data set, we built a truth set. So assuming you have two groups of people, big data set, you have healthy group and have the COVID-19 group, okay? So to define the false positives, what we did, we split our group into two. And what I mean, the healthy group. And then we compared those two healthy groups. I'm sorry, I've split one of this person into two, but well, here we go. And basically, we do differential expression from that group versus this group. In a perfect tool, there should be no differences. Because the differences are random, there should not be difference. So this is one method to kind of build our false positives. But how can we basically get the true positives? So the way we did it, we know that COVID-19, and we also used rheumatoid arthritis, we are pretty sure that some go term, some pathway must be hit. It's just, we know that, not all of them. So basically what we did, or this is where Dam came in, our immunologist, who is leading the paper, and he basically said, I know from my immunology, I know all the go terms I would expect with high confidence in a comparison of healthy against COVID. Hence, I know all the genes that should be part of that. And these genes, they form our truth set. And then we compare that with a differential expression against that. And the overlap is then the truth set. Is it perfect? No, it's not perfect. But this is a good approximation. It's the best we found. So putting that, so now let's first to talk about the false positives. So we did, as I said, two studies with at least 7, 8 replicates. And the first thing we looked at this number of p-values in the different methods. So what you want if you test the two healthy groups, you don't want the p-value to be significant. You don't want them to be zero. So in the first two lines, you see single cell MUST and single cell DSIC2, which are two single cell methods. There are ways to tune those. You could do Latin variables for MUST. We didn't do that because you took it the way it was in the um, in the tools like Surat, the way we used it. And then if you compare it with MUST, and it's a different way we sum up cells, it's not really important. You can see we still have some false positives here, but overall it's mostly one. And Armin DSIC2 is even better. So those tools control best our false positive. And no surprise, you can see that the pseudobulk DSIC2 is actually best. It has nearly no one, but what's also nice, it has a kind of range of p-values. It's something you would expect rather than to be something like Peggy. So on the false positives, I would say, like pseudobulk is still kind of good. Then we looked at different tissues. So here, basically you see different clusters we did from the COVID, and the next three are from the rheumatoid arthritis, different clusters. And then here you see the different tools. So the first four are cell deep, and I don't want to go into too much details, but there's different ways you can change. You can change the pooling strategy. Is it random? Is it a k-mean? You can change the tool you want to use, the seek or must, and you can change the way you do you sum up the meta cells. So you want to sum all the counts into one, or you want to find the mean of it. And interestingly, if you look, the best tool we have in terms of false positives is actually the cell deep Armin D-seq2. And I would say that is very, very similar to the D-seq bot. Importantly, these tools are under the 0.05 false discovery rate we set. But you can see that in the single cell methods, especially MUST, really overpredicts, and also here single cell D-seq2 is too high. On the bad side, you can see that some of our cell deep implementations also don't perform very well. So the conclusion here is that the single cell methods overpredict. I would say first, our framework is kind of working. The single cell is over-predicting the false positive. Pseudobulk controls it very well. And CellDeep can be good. So our tool can generate good results. Then the next test we did is basically true positives. And you can see here that overall, we can see that the single cell method generate a lot of basically true positive rates. However, our CellDeep is very close as well over most of the tools. The only one that's not so good is the NK cell. And the reason is that we had very few cells to pull. So we didn't have so many meta cells. So overall, I would argue that if you take it together, then it looks good. However, how can we visualize this data a little bit different? And then we came up with this kind of heat map view. So on each row, you see basically a tool here. So this is basically single cell MUST as above. Then we have the pseudobulk. And then basically we have our different cell deep methods. And green means that so we have, I think it was 100 Go terms to be found. And then every green sees it found the Go term. But obviously, it's always the thing, what's the balance? ROC curve, sensitive specificity. So to kind of capture that, you can see here the number of differentially expressed genes. You can see the number of found Go terms, and then you can see the ratio. And here I would argue that CellDeep has the top ratios of numbers of differentially expressed genes and Go terms captures. So overall, if you use the right data, the right parameter sets, CellDeep can be very good. The only crook is that you don't know the best parasite a priori. So the conclusion is that differential gene expression in single cell is still tricky. If people want to tell you something else, it's not. You will find genes. You have to be taken with a pinch of salt. And obviously, super strong log fold changes will be different in most tools, but the tail end is very tricky. So, and here we propose actually that CellDeep is actually a good balance and is a valid tool. So that was basically one way we can quality control our parts. This should be soon, should be out. And this is a collaborative project from my lab. And but the question is now, how can we go back? So should we go back to bulk RNA-seq? So as I said, your single cell is tricky and bulk RNA-seq is super well controlled. So imagine the following thing. You have a parasite and the wild type. In 1 hour, it goes from this development state to that development state. But then you generate a knockout. And without you knowing, due to the knockout, it grows slower, half the time. So when you now do a differential expression, you're obviously comparing wild type 2 with the knockout 2. However, you're comparing the parasite in completely different life stages. So it's a little bit like comparing me with you guys. You have most of you are half my age. So you should be far more energetic and you still have a lot of hairs and no gray hair. So, but then what we want to test, I mean, the test was maybe I drank 5 liters of water every day. But this effect you wouldn't see because the effect of the age would be so much stronger. So the problem is often if you have that in some, especially processes who follow pseudo time, we basically don't know where our bulk sample is. So that comes very close to deconvolution of pseudobulk with single cell. And this is a little bit the idea. What is it? Basically are tools where you have your single cell data set and you use there, you do the classification in different classes. And then you put in your bulk. And then the deconvolution tool tells you how many of the frequency of the different cell types you have. And there's CyberSort, very much known, DVLS, and other one is music as well. But none of them works on linear process. All of them are of discrete cell states. So with that, basically, we thought, let's do following. Let's do it in a linear process. And the tool called Blaze or Blaze, depends if you're more like a French-speaking person. You do first, you take your single cell and you basically order your cells along a trajectory. Then you bin your trajectory and put it into bins. So next, you take your bulk RNA-seq sample, which is always a mixture of different cells, and you basically compare the correlation between of your bulk with the different bins, and basically you take the one where your correlation is the highest. And that's basically where you place your bin. We did, if you read the bioarchive, we did a bit more of statistics, or Andrew did this, and who is doing the tool, it's his project. But basically important is we can use it. So here, for example, we have bone marrow hematopoiesis. He took a T cell and B cell trajectory. You can see that here. And then basically he built this with slingshot. And you can see here he has the different bins, and the different bins are parts of the different life state, or the development stage of B and T cells you would expect basically in the bone marrow. And then he took a bunch of bulk RNA-seq data and mapped it back to his trajectory with Blaze. And you can see, basically, you can see the diverse parts of where the sample sits. You can see also nicely we have a bit of, so the darker it is basically the more correlation there is. And often you can see that bulk never really is just one state, it's always a development process. So this is the way we use Blaze here. We also use it in a parasite content. So this is, we took data from a work of epicoplasm of heat. Basically, it was John Adams, and basically they had a piggyback. So basically it's a library where they can generate very efficiently mutants and knockouts. And they found a mutant, LL5R mutant, that was basically sensitive to heat, and they did bulk RNA-seq. So we took those data after it was published. Andrew then first built the trajectory, and then he took the normal NF54, which is our canonical reference genomes, and under normal conditions, you could see he took basically the data, and you could see the correlation was highest at the bin number 3, and under heat shock, it's bin number 4. So you can already see that here we have a temporal difference, right? Now, if you look at the mutant grows definitely differently to the wild type, and under heat, it's also it's quite different between the PB6 normal condition and the heat condition. So, and without having a check where what happens if you then compare, for example, these data with these data. So these are mostly bin 3, 4, and these are mostly bin 1 and 8. So we're using Blaze now to try to correct this fact that there is a different time in your bulk. So basically one we said is 1 and 8. So let me just find it, is somewhere here. And the other guy is already 4 and 5, which is somewhere here. Oh, I'm colorblind. We should use different color. But you can see they're quite far apart. So what our tool does now, it basically gets the bulk RNA-seq data, the list of differentially expressed genes, but it does also do a differential expression of this spin versus this spin. So basically it compares 25-year-old with 50-year-old. And then it looks at all of these genes that are differentially expressed. And these are those, 282. And then basically we subtract those, from our bulk one. So rather than having 340, we just have 203. And then we say, yeah, well, less genes now. Well, if you now look into the heat map, and it's a bit difficult to read, but what you have different comparisons. Here you have the Blaze differentially expressed genes. So basically the bin. Then you have here the bulk, my mistake. Then you have the bin, and here you have the corrected. And here you have different go terms that are significant. In blue, the one they have. And you can see that significant go terms are quite different between the bulk initially and the bulk corrected. And overall, obviously, it's heat, and I want to go too much into details, but you can see overall you have vehicle docking, you have import into cell, and other biological processes which are very well explainable through the heat rather than just the different life stages. So overall here, we shown that you can use Blaze to actually correct bulk and seq. And one thing, there are different methods to do that. Obviously, there's linear part in malaria, but you can use it for every pathogen, for every growth. You could use it for cancer, etc. And obviously, you can correct it, but it's also powerful to just place your bulk RNA-seq on a trajectory. And that's Andrew. And here, basically, you see the link to the bioconductor if you want to try it. How much more time do I have? Roughly 10 minutes?
10 minutes, yes, at least.
Perfect. Good. So that's basically Blaze. And now coming to the last project, and I start a little bit, I don't know if you heard about it, but very excitingly, there's these new technologies, which is called single cell platforms. And what we're interested, we're not just interested basically in parasites, what they do, but also what they are. So where they are in the brain, what they're doing in the bone marrow, how they're cyclotested, how is the microenvironment enrichment? So what is the heterogeneity here? And there's tools like Xenium and Cosmic, and now also StereoSeq, where you can basically generate data, spatial data to the single cell resolution. You can basically, here you have your slides in FFPE, you basically put your single cell, and then you can afterwards build your cells together and do analysis. It's working. It's prose-based, so single cell, you always need viable cells. Here you can put it in a block and then do it. Overall, you have 1,000 to 6,000 probes, so this is the good news. And you can also do extra probes for pathogens, for example. The bad thing is, and there's NanoString and 10x, and now also the BGI sister. The bad news is it's not as good as it sounds. So if you look at some of the technologies, so they say there are 6,000 probes. We just get between 80 to 300 genes per cell. It's very difficult to do the cell segmentation because if you think basically this is two cells, but they are together in the Z, they overlap. So how you can do the cell segmentation. So albeit this is so difficult, you can still, and not resolved, you can still already get some very nice um, results out. So what we thought on the long term, I'm interested in parasites, into how they're going to shake up the environment. So before having any data, Adnan, who was a postdoc when I was in Montpellier during my sabbatical 2 years ago, he said, well, why don't we do a way to kind of look for niches where we use entropy? What is entropy? Entropy is basically something that looks for order or structure. So if something has a lot of structure, then it's a low entropy. If something is in disorder or a lot of uncertainty, it has high entropy. And the idea is as following, assuming this is your spatial biology, assuming you know that G1, G2, and basically here in these different parts, you have pathogens. We want to understand that in those regions, are there any different frequencies between the cell types here than in the rest? So if it's low entropy, we find more we find more signal. That's the math, and we came up with this double or well, Adnan came up with the help of a video of this entropy RC tool, and it's a double sum because he needs to compare. That didn't exist, but anyhow, soon it should be on bioRxiv. And because we didn't have any data sets, we basically looked at an Alzheimer data set from a mouse, it was a mouse model with plaques. And basically, you can see here the plaques, and around that, basically, he drew, or the tool drew basically the niches. And then he compared basically the frequency of the different cell types within this niches, and used this entropy. And what you could see, basically, here you had a lower entropy and these cells, and without going too much into details, you could find some interesting signals of some cell types are more relevant for Alzheimer, and that will mitigate basically to the plaques. And what you can see here, it's a number of occurrence overall. You find them, but obviously the ratio of your regions around plaques is far smaller, 8% of the tissue versus 82%. So if you do that, you can really see there is signal in terms of enrichment of our cell types around the plaques versus somewhere else. So here we go. So obviously, luckily we then had a data set. This is what you're going to see. It's a collaboration. So it's a different pathogen. It's basically we use a juvenile schistosoma mansonia, who is a worm, and basically infected it in mice in the lung. And then basically the xenium was run with parasite probes. It's a collaboration. So now Dr. Olympia Hardy was a former PhD student of mine. She organized everything from getting a good quote in and organizing all the experiments. Gabriel, who's a former colleague from Sanger, and Matt, my former boss, actually was now also in Glasgow. They helped with this project. And basically what we had basically fixed lung from mice, and basically the titer slide, and then put it on the xenium. So what you're going to see is the xenium plate. And then the question was, as we did parasite probes, you can put up to 50, can we find a parasite and can we find structure? And the fact is, yes, we can. So you can see here, this is the lung tissue of one mice. And we zoomed in. Here you have the different lung cells. And in color, you can see the worm. And not just that, you can see also the different annotation of the part of the worm. It's not finished. If you zoom in, we could see that the nucleus is far smaller. So it's more fine-tuned, it's a bit more tricky. But she also found other ones, so you can see here how they are. And now, obviously, what we're interested is to see what Olympia did. Is there any difference between the immune cells around the worm than somewhere else? And sadly, there is not. So somehow, the schistosoma worm in this stage can traverse undetected through the lung. And sadly, we already knew that. So, because it was reported like 30, 40 years ago, apparently, I'm not a one person. So we confirmed something we know. But for me, again, I'm interested in tools and technologies. The good thing is that you can actually detect parasite in worms with the probes. So with that, I'm finishing. So basically, Adnan's tool will be used for that. If you're interested in the GitHub, sorry, you can um, sorry, you can download it here. And this is a paper to come out soon. With that, overall, I hope I give you an idea of the tools we do, that we have hierarchy of regulation, heterogeneity, and we try to dissect it with different methods. You can see there was different types of methods we use, from like machine learning. I wouldn't call it AI yet, but just to straight programming. Often the problems are not necessarily the tools, it's mostly to get the data in the right shape. And very important is that there is highlight there's some really limitation of current methods. With that, I would like to thank all my team. Some of the people who moved on, like Aaron was a master student and is now working in London. Olympia did her PhD recently. Adnan was during my time, and now obviously thanks also to my lab in Glasgow and my new lab in Hamburg. And this is actually a photo from a retreat we did 1.5 years ago in Montpellier, South France, with rain. And I realized I need a new retreat for a new picture. With that, just if you're interested in bioinformatics, this is actually our Hamburg summer school. You might want to visit if you can. But with that, thank you very much for listening.
Thank you, Dr. Otto. That was fantastic. And you just explored a lot of topics, a lot of different tools. And as I was looking at your presentation, I realized that a lot of the things you are working on, different labs and different students from IOB have been working on them from different perspectives, I guess. I expect a lot of questions to come in. And if any of you have questions, students or faculty, you can just unmute yourself and ask, or you can write it on the chat and I will read it out.
Thank you.
Anybody? Okay. If nobody else is coming up, I have a question I would like to ask first. Um, I was seeing that in most cases, you were working with immune cells or data sets from immune cells. Um, do they, when you are using those tools in other types of cells or from even other organisms, do they still have the same level of efficiency? Especially if you're like training them for immune cells, does that translate on to the other data sets?
So I guess so around single cell deep, and Blaise, I would say yes. The problems is not necessarily dependent on the type of tissue. It's mostly how you sum it together. We did try some of them also on malaria with similar results. However, sometimes you always have some odd ones. So just to say it always will work, it's not necessarily. So for example, if you do single cell, you need very good annotated UTRs. So this is something you have to be aware of. And sometimes you have less genes, but I think overall, all the tools we have, OpsML obviously not, is really specific to malaria because they have this sequence. But you could use, I mean, in the end, it's just a classifier for that. And I guess the spatial transcriptomics part will work mostly on mouse and human. But for any types of cells, there are different panels. They have immune cell panels, they have cancer cell panels. So as technology, they also work, but it's limited to those two species because this is where they make money. However, if you want to do other species, you could use StereoSeq or 10x Visium, who have a sequencing-based approach. Does it answer your question?
Yeah. Thank you so much for that. Anybody else can unmute or you can also like send React and I can unmute you if you want. In case nobody else is coming up, I also had a different question. It's not about the research per se. You were saying that anybody who's an academic needs to move every 5 to 7 years. That's some what anything. Well, is there a reason for that philosophy? You that like you develop this philosophy rather than like staying somewhere and building a career out of there?
I think it's mostly a gut feeling. A lot of people did it. I guess it's often because you have to move rather than you want to move. So I think it's important. I personally think it's good to learn different ways of working because there's not just one way. So I guess always say after your PhD, you should change the lab to learn a different culture. And then it depends a bit on how it is. I think there's no rules as such. It's just that what I feel from personal me is that after some time, routine kicks in, and it's different from time to time to basically get something different from the environment. And changing environment helps you. There's also the question of getting stuck. So I wasn't able to get promoted further at Sangha, so I needed to leave Sangha. And sometimes you just get maybe a better position somewhere else because they really want you, and where you are, the undervalue, or sometimes it's. But on the other side, I guess if you're super happy where you are and you're close to your family and you like the spot, there's no need to change. I don't say I wouldn't judge people that they stay. It's just that I think I like to move. So I like different environments. I like the fact to build new relationships, to start new projects, learning new techniques. And then basically for this move, my final move, it's also I think from the package, it was very good for me. And it has also part that in the end, I'm German, as you said before, coming home. I was a long time just 150 kilometers south of here. Then I study very close to here. So it's also the coming home. And I feel a little bit have traveled the world, learned a lot, and now I kind of give it back. So it's a lot of things. And so far, I didn't, although I loved Glasgow a lot, this fantastic city with a lot of very nice people. Obviously there is the gray winter and the rain. There's no shying away from that. But I also had a very good time there and did good research.
Thank you. I think like for a lot of us, like um, graduate students as well as postdocs, we actually need to know like what decisions we need to make and in to further our career and like find the correct places to go to where we can find those opportunities. I'm not seeing any questions on the chat. Is there anyone who wants to add something? Yeah, Anthony, you can ask anything you want.
So first off, thanks, Thomas, for joining in on us today and giving that awesome talk. I had a question about Cell Deep. Now, um, you mentioned some of the limitations of the UTRs being problematic. Did you test any like different technologies? So 10x versus SmartSeq, and do one of these perform better or worse in terms of predicting differentially expressed genes, or let's say missing differentially expressed?
Yes, it's a very good question. We haven't. So we mostly worked on 10x. I would say that SmartSeek 2 and 3 would be slightly better because you have a deeper coverage. So you have a lower dropout. So Yi and Dom, they did some tests and look at dropout rates. And basically, the less dropout you have, the better the model is. And the other thing is that if you look in your, obviously there's some genes that are high expressed, but a lot of genes in 10x that's supposed to be expressed, they have two UMIs. That's two individual transcripts, and some have one. And then and this could be just noise. And in SmartSeq, you don't have that. In SmartSeq, although you don't have UMIs, you just have reads. So I think in terms of I wouldn't be too worried. But the thing is, you have the framework, and you could just compare it, right? So you could. But on the other side, with SmartSeq, you just have a 96 384 plate, and so you also have less cells. So I would just say that in SmartSeq, every of your cells already a meta cell.
Okay, great. Thank you.
Thank you, Anthony, for that question. Anybody else, if you can jump in? You can just unmute yourself and ask. Otherwise, we actually are at like kind of at the end of our allotted time. So I would thank Dr. Otto again for joining us so far away. Hopefully, we'll be able to meet you sometime here or in Germany or somewhere else. Let's see. And I believe the tools you have been developing will end up using it like I am using one and sometime or the other. Thank you so much for joining us today and have a great day.
Thank you for having me. Thank you very much. Bye.